Yes. How are you doing? Good. Good afternoon. Good evening.
We're doing okay.
So I think I'm not sure if you can even be able to hear me because uh, I've made my computer free. I did not connect it to any speaker so that on your side you can be audible to me. If you can hear me, that's fine.
Yes, we do hear you.
Yeah. Okay. So this is how we are going to do it. So it's not like it's not going to be a lecture. It's not going to be a lecture, so you can you are going to unmute yourself, especially if on your end there's no any background noise, so you can feel free to unmute yourself, unless maybe there is a noise around. So maybe you can be unmuting and demuting accordingly. So my recorder. My microphone is on. My audio is on so that we can interact because I think there is no noise where I am. So the way we are going to do it now, because this is just going to be like a revision. So it's not like we are going to revise everything now because we may hold the same session next time. So uh maybe so what we are going to do, we are going to start with one course, and then uh, you will suggest which topics you want us to revise. Then I'll be taking you through the key points in that particular topic in the course. So I'd like to ask which subject you would like us to begin with, and which topics do you want to us to talk about? And we will be sharing what we know about the course, which is going to be helping you to in your reading plan and in your reading target. So one can suggest which course would like us to start with, and if there is any topic in that course, so that we can try to talk about that one.
Yeah, that's sorry, Anna. Do you hear me?
Yes, yes.
I would like to suggest microbiology.
Okay, microbiology. All right, so microbiology. Like which topic specifically can we talk about?
Specifically, I would like to focus on uh, MTBs. If,
Okay,
if
tuberculosis.
yeah,
That yes, like mycobacteria. So mycobacteria is what we are going now to talk about in microbiology. So I think everyone has read or attended microbiology class. Maybe you may not have covered already the entire course, but at least uh, at least you we covered it. Like I mean, maybe last like a few months or a few weeks. I don't remember when we started this course. So let's try to talk
And
about.
and
Mm,
and I think if there is a easiest way of approaching biochemicals, they are so confusing. Like
biochemicals for mycobacteria,
like for what number of organisms?
all of them.
Yes, yeah. Like, Hmm.
Okay. Yeah. So which means that now our focus now is going to microbiology, and yeah, from there I'm going to guide you how you are going to read microbiology, especially what do you need, especially because microbiology is very wide, and even in exam it is very wide, and as you say, chemistry part of microbiology or microbes, yes, it can be very confusing, but it is not actually confusing. It is not so confusing because we first of all classify it, have we put microbes in classes so that you can do, because when you put them in classes, they have their classes, they have similar biochemical reactions. For example, if you want to classify gram-negative bacteria, all those bacteria they have almost similar reactions. For example, there are some tests you have to confirm for all gram-negative bacteria. We can say oxidase, for example. We can say pure test. We can say metal red. We can say VP test. We can say like ONPG test, urease test, and DNase, for example. Though DNase is not so common for every gram negative. Those tests. So how you are going to approach them is that those are the tests for all gram negative. But you have to understand that, for example, if you take lactose, for example, lactose fermentation, that is about for like fermentation and oxidation. If you take lactose, if you want to focus on lactose fermenter bacteria, then it means those bacteria must be in a group of Enterobacteriaceae. That is the family. Because you cannot think of lactose fermentation and try to focus on Vibrio, for example, Moraxella, or even Neisseria species. They don't have anything to do with lactose fermentation. So lactose fermenters are very few. Others are none lactose fermenters. And if bacteria is not lactose fermenter, then it is going to be either non-lactose fermenter or delayed lactose fermenter. So when you take non-lactose fermenter, you have a few in Enterobacteriaceae because as you remember, Enterobacteriaceae or enteric bacteria, they represent a large group of aggregated bacteria. Then you have to pick some of them that are strong lactose fermenters. For example, Klebsiella is a very strong, and Enterobacter, I mean, and E. coli, Escherichia coli. So those are the strong ones we have. Others, most of them are not lactose fermenters, like Salmonella, like Shigella, like Proteus. All of them, they don't ferment lactose. So that's an example, like how you can try to classify, because out of none of them can ferment lactose. Some of them can be like delayed because they can test for any PG positive. Otherwise, none of them ferment lactose. So then if a bacteria is a gram negative and a bacteria is a non-lactose fermenter, so what you have to do in order to also try to minimize or to remove a big number of bacteria to choose from, that is the test that you must do for gram-negative bacteria. Oxidase. So oxidase is a must, the same as lactose. So when you do oxidase, it means you want to make sure that a bacteria is either in Enterobacteriaceae, if it was lactose negative, and then you are going to check oxidase to confirm whether this is in group of Enterobacteriaceae, because all of them, no one is oxidase positive. All of them are oxidase negative. So then you have made that choice. Oxidase is going to be negative for all Enterobacteriaceae, except what we call Pseudomonas shigelloides. But Pseudomonas shigelloid is also not, now is not classified under Enterobacteriaceae, it is classified as a Pressiomonas species. Pressiomonas, because we have Pressiomonas species, Aeromonas, Campylobacter species. So it is somehow outside, but it was classified under Enterobacteriaceae before. So that is the only one that is oxidase positive. So then it means if you see oxidase negative, it means that bacteria is within Enterobacteriaceae. If a bacteria is non-lactose fermenter, it means you are going to think of Salmonella, Shigella, Proteus, Providentia, Edwardsiella. Those are the ones that are non-lactose fermenter, but negative for oxidase. So then, if you see that one, then you're going to check. You're going to check another possibility, for example, nitrate reduction, and then you may also look at DNS, for example. Those ones are also obviously DNS negative. So that is how you approach them, and you check all those characteristics. You can be able to choose. So maybe let me take you through that one. Then we'll look at some questions and see how we can go about that one. It is very simple. Like microbiology is not so difficult, but of course what you have to do is to try to design your own classification of bacteria so that you exclude all those that are not reacting the way you see it like that. So because even if you use like TSI, triple sugar ion agar, TSI, even if you use TSI, some bacteria, for example, TSI, it is about fermentation. That is the fermentation for TSI, triple sugar ion. So because it is a TSI, fermentation can be glucose, can be lactose, can be sucrose. But what you have to know, all gram-negative bacteria are glucose fermenters, except what you call non-fermenters. Non-fermenters, we have them like, what you call um, sometimes I forgot those kind of names. So the non-fermenters, let me recall some of them. I know them, but those names sometimes they used to confuse in my mind. So those ones we have them in Pseudomonas species. Sorry, Pseudomonas species are non-fermenters. We have Pseudomonas aeruginosa. We have the Pseudomonas putida in their groups, and Stusseri, all of them. The Pseudomonas species are not, uh, even Agrigobacter species, all of them are called non-fermenters, so which means they don't ferment glucose at all, but some of them, they oxidize the glucose. So which means if they want you to mention Pseudomonas species, you have to know that you are going to see something called OF there. OF. So can someone tell us what is OF? OF test. And how do we interpret them? How do we do? How do we perform them? OF test. Teza, can you tell us what is that? What is OF test? And because OF is one of the major biochemical tests you can do for in microbiology, OF test. OF. Esther, do you have any idea about it?
So
OF.
OF test.
Yes, OF test.
One you should displace an oil on top of it.
Another one, you don't put oil. Okay. Yeah, how do we what is the principle? After adding oil and that one you don't overlay. It is called overlaying oil. That's one that you don't overlay oil. So what's what's the principle that
Two test tube, one with oil and then the other one with no oil, and then you what do you call it? Oh,
like what is the principle of that reaction?
that's the idea.
Someone else, let's try to interact and discuss. Because the reason why we are very few here is because we have already read and studied this, and we are getting ready for exam. So even though not all of us are here, but I've already sent them. I think like we have like other two people or three people here who are not ready. I think we are all here. Oh, let me see. Are we seven here? Yes.
What are the bacteria? Is a fermenter or an oxidizer?
Yes, oxidizer and fermenter. Yes, Angela is there. And they're like, please talk to us, no problem. You can even mute yourself and express yourself. You don't need actually to even to request.
Okay. So thank you very much. For OF, I think it is like oxidation and fermentation, where we get one tube, we do not seal it, and another tube, we seal it with oil to cut oxygen supply. So this tube where we've not cut off oxygen supply, if the organisms that are there utilize oxygen, I think we are going to have a reaction. But in this tube where we put oil to cut off the oxygen supply, even if they require oxygen for what? For their survival, a reaction is negative because we cut off the oxygen supply. I don't know whether it
Yes.
will.
So which chemical substance that is there that bacteria are using, whether they are in oxidation tube or fermentation tube? Because I'm looking for that particular content. So sorry, just tell us, then I will add something.
I only remember the indicator.
Yeah, so now indicator is of course pH indicator. So now okay, let me just briefly introduce this particular test. So what happens? We have what you call OF medium. OF medium contains glucose. First of all, glucose is the major content of it, or it contains glucose. So because in order to test OF, it's like, first of all, a bacteria is not fermenter, or a bacteria, if it ferments but not lactose or sucrose, but at least the bacteria can be able to ferment glucose, or a bacteria is non-fermenter at all. So how do we confirm either it is not fermenter or it is only fermenting glucose? We have to use OF test. OF test contain glucose at high level, like 1% of the medium is glucose. So 1% of medium is lactose when you are doing TSI, triple sugar agar. So now here what we do, because we want to provide, first of all, oxidation medium, which means glucose must be there. Glucose must be there, but also oxygen must be there. So that's why we don't overlay oil on it. Like OF media, we put in the tube and we don't overlay oil. So let me see if I got that one. The particular, yes, I think I got it. I want to show you like how the tubes look like. This one. I hope you can see my screen. Right? Can you see?
Yes, we can see that.
Okay, you can see the tubes here that are yellow. So you can see what is inside there is carbohydrates, but specifically we have glucose. Glucose is there. Glucose must be there, and it's the one that is in each tube. So it contains 1% of glucose. Then it also contains 0.2 peptone. So, and of course, indicator. Indicator is bromothymol blue, which is the indicator in this tube, bromothymol blue, but not metal red. Not metal red. It is bromothymol blue. That's why it may look like this when it does not react. It's not like TSI, which contains pH indicator is metal red, but this one contains only bromothymol blue, which is a pH indicator. So what happens? You have to have always you perform the test with the two tubes. The tubes are in a set. We have set 1A, 1B, 2A, 2B, 3A, 3B. So each tube with its own microbe, for example, with its own sample. Tube number 1, tube number 2 with one sample. Tube 2A, tube 2B, one sample. Tube 3A and 3B, one sample. So we have maybe 3 patients here. So what we do, this tube, you can see it contains oil. Oil, that's why there's a gap between here and here. We have overlaid oil. Oil need to just put oil and we close tightly. So the reason why we are adding oil is because we want to close very well so that there's no entrance of oxygen from the top, and you have to tighten the cap, this cap, and we keep it like that with the sample inside. But this one without oil, we don't close it so tightly, but we keep it loose so that some amount of oxygen can enter this tube. So because we want to detect any possibility of the oxidation. So then the same for the second set of tube and the third set of tube. So if bacteria is able to, because normally when the sample, when this tube is not inoculated, it looks green, but if there's any amount of oxidation, if there's amount of oxidation or fermentation, it changed color to yellow. So because there is a pH indicator of acid, a very small amount of acid produced. So for example, if bacteria here, the reason why here is yellow, it means that the bacteria that is here is a fermenter, the same as is a fermenter and oxidizer. Is a fermenter and oxidizer, which means a bacteria now, it ferments and oxidizes. It means it is facultative anaerobe. It survives in anaerobic condition, the same as aerobic condition. For example, Escherichia coli can show these reactions. It means Escherichia coli, because it's a facultative anaerobe, it can grow in anaerobic and ferment glucose. It can also grow in anaerobic and oxidize glucose. So because otherwise without oxidation, then there is fermentation. So the second tube now here, it means that the first tube where we add oil on it, there's no oxidation, because there is no fermentation because the tube remains green. There's no fermentation, but there is oxidation. So which means this tube, bacteria that is there, it is non-fermenter, but it is oxidizer. So it means this bacteria, it is only oxidizer, but not a fermenter. So which means this bacteria is not able to grow in the absence of oxygen. So it is called non-fermenter, but oxidizer. For example, what you say as Pseudomonas species. Pseudomonas species are non-fermenters, but they are oxidizers. So for the last set of tube, we have there's no fermentation, there's no oxidation. So now a tube is like, this bacteria is like inert. We have some inert bacteria. We have some inert bacteria in Aggregatibacter species. So which don't do anything. They don't ferment. They don't ferment. They don't oxidize. Even if they just grow. Some of them, they grow either in absence of oxygen or in the presence of oxygen, but they don't have enzyme that facilitate them to ferment or to oxidize any carbohydrate. So they are called asaccharitic. Asaccharitic. So some bacteria, we will see them, and when you are reading them, you will see, they will say the bacteria is asaccharitic, which means it does not lyse, does not break down carbohydrates. It is called asaccharitic. So it is non-oxidizer, even non-fermenter. That is how you can know it. So which means, apart from, if you are talking about all gram-negative bacteria, all of them, we have two groups. One group is that are those bacteria that ferment lactose. We don't need to bring them here because we use TSI for that purpose. We use the TSI. If we are dealing with lactose fermenters, we can use TSI. It is easier because TSI contain lactose, but OF mainly contain glucose but not lactose. So we can use TSI in order to study those ones that are lactose fermenters. But if you have non-lactose fermenters and you want to distinguish them from non-oxidizers or non-fermenters, so we have to use the OF medium. Yes, so that's what you can do in our small classification. We're trying to classify gram-negative. Okay, so that's what we call it. So, and the purpose of TSI, TSI is we want to know if a bacteria, first of all, there are like four major reasons we want to test here. First of all is lactose fermentation. Secondly is a non-lactose fermentation, but glucose fermentation. And thirdly, we are going to test production of gas and then production of hydrogen sulfide. So for example, which gram-negative produce hydrogen sulfide and is a major characteristic? Who can remember that one? Can remember and tell us. So which bacteria that produces hydrogen sulfide to be its major identifying feature is Salmonella, isn't it?
Yes.
Correct. That is Salmonella, because Salmonella, when you grow it on any media, like for example, because production of acid just is that like blackening of it's a blackening of medium, whether it is a solid medium, whether it is semi-solid like this, slant medium, so it is a blackening. So, and Salmonella is when it is, for example, when you use like hecton enteric agar, it is very obvious. HE, hecton enteric. Hecton enteric agar. So how we will identify Salmonella is the blackening of medium, just around the colonies. They see some black spots there because of hydrogen sulfide production in hecton enteric agar. So that is like one of them. So, but when you have the TSI like this, though you may find the question where they ask you to interpret, but the major purpose of TSI is, first of all, to study bacteria that are not lactose fermenters or sucrose, because most of lactose fermenters, they also ferment sucrose because both of them are disaccharides. Yeah, but we have also another group that don't ferment lactose, but at least ferment glucose. For example, this tube number 3 here. So because you see, there is a blackening. I mean, there is a not blackening. There is a yellow color produced here in the butts, but not in the slant. Slant is still red. So it means only a small amount of gas, a small amount of acid was produced in the butt, but in the slant there's nothing. So which means this is a glucose fermenter. And remember, glucose fermenter is any other gram-negative bacteria apart from Pseudomonas species, but also apart from the major lactose fermenters like Klebsiella. Even a tribe called we have tribe that is called the Klebsiella tribe, and even Escherichia tribe. Most of them they also ferment lactose, but except of course Shigella, because Shigella is also classified under what we call uh, Escherichia tribe. So they are not similar, but they are classified under one tribe. So okay, that's it for the TSI and the KI. They are the same things.
So, I have a question.
Yes, please tell us.
So, in TSI, can you also tell that this bacteria is a fermenter based on the slant or it's a oxidizer? Because the slant is exposed to air and the bacteria is not exposed to air. So, if it is fermentation down there, I think maybe try to make it clear with me. Like,
Sorry.
I'm confused with this part because
Yeah, tell me. Let's just remove confusion. Tell me again. You say?
can you tell that this bacteria is a fermenter or oxidizer on TSI based on the air exposure? Since the slant is exposed to air
Oh,
and the bat is not exposed to air, it's covered like.
covered.
Mm.
Mm. Oh, yeah. I got the point. So for TSI the game is different, very different for TSI. So what the difference between this and TSI? I mean, difference between TSI and OF medium. OF medium, we're talking about closing and opening, and adding amount of oil on it to cover medium and or not. But what makes it a difference? Uh, what makes a difference is that when we are incubating TSI, there is amount of peptone. Amount of peptone is very high. So even if a bacteria is a oxidizer, it cannot turn TSI yellow. Yeah, because in oxidation process, glucose is the one that is oxidized to produce amount of acid required to make the medium yellow. But you remember in TSI, the amount of carbohydrate, the amount of uh, glucose that is there is very
0.5.
low. Is very low. It's like 0.1%. It is very low. Even if there is oxidation going on in the slant, it cannot become yellow because the amount of acid produced will be very low. And of course, as you remember, is that the glucose fermentation that is happening in the bat, because in the bat, of course, there's no oxidation. In the bat, there is fermentation. If glucose fermentation is happening in the bat, the amount of acid produced from that glucose is also very, very minimal. That's why the yellow color produced in the bat is not so enough. It's very, very, very small. Like that. That's why you see here, tube number 3, it doesn't look like tube number 1. The color of tube number 1, yellow color, is very intense, but here it looks between red and yellow. So because of amount of acid produced here is very small, which is not turning the bat so yellow like the previous one, like this one. But that amount of acid produced upon exposure to oxygen here, or even if there's no oxygen exposure, even if there is just acid infiltrating towards the bat, still towards the slant, but still the slant can't be yellow because of peptone. Because remember, a bacteria which ferment glucose, it also uses peptone as a source of carbon. Peptone is also produce carbon. So when peptone is being used after exhausting glucose, peptone is not peptone after peptone use or peptone catalysis is going to make it alkaline but not acidic. That's why for this tube, even if you have to wait for like you have to extend like 5 hours, even this bat is going to become red again because of peptone. Peptone is going to be used because the glucose was exhausted already. And this one, why it is very yellow is because peptone is there also, but peptone was not touched because amount of lactose and sucrose that are here that are being used by bacteria are enough for 24 hours. But when you come to test after 24 hours also, obviously, if you come to test like maybe at 27th hour, this tube will look like this. Will look like this one. Because when amount of acidic is exhausted, the tube will revert back to this color, to this red color because of peptone use. Because when bacteria finish carbohydrate, it uses peptone. That's why also here it is like this. So amount of glucose was exhausted, and then peptone is now under use. Of course, when it is like when you delay, of course, the peptone alkalinity will be higher than the acidity that was produced in the bat, and the tube will turn back to red color. So that's what will happen here. So having slant does not give it anything, anything that can make acidic condition to be produced in the slant, because still glucose is very low. Secondly, we have a large amount of peptone, which will not allow the acid to be produced in air like this one. Yeah, because obviously you know that amount of acid produced by oxidation is very low. Very low. It's not like fermentation. It's very low. Still acid is produced, but it is very, very low in concentration. Yeah, that is about the TSI. It works like the KI. I don't know if I try to make it clear. But of course, what you just need to here to understand is to understand the principle of trichosugar agar, and what are some of the challenges that are happening when it comes to delay in reading too early. For example, if you read too early, like less than 12 hours, even here, the slant will be yellow. Still, after less than 12 hours, the slant will be yellow also, even if it is only glucose being fermented. So those are like some challenges that you'll find when you are reading TSI. And if you delay to read, things will change completely, and you will not even revert them. You will not know what's happening. So, and also, of course, displacement because of gas. Some bacteria produce gas. We have them. Those gas producers also. Some bacteria produce gas, others don't. Some bacteria produce hydrosulfide, as we gave an example of salmonella, and then others don't produce hydrogen sulfide. So you'd see them, for example, Escherichia coli doesn't produce, but Klebsiella produces hydrogen sulfide. So those are some of the reactions that maybe you can try to memorize, because in some cases when you are reading this, you also have to memorize something so that you know the key points. For example, talking about Escherichia coli. So what are the major characteristics of it? In order to know that, of course, it's very good also to read about this stuff. For example, this one is called what? Or NPD. NPD is very simple. So, okay, tell us what's the principle of NPD, and then we look at the principle of natural reg and VP test. So they are the same thing. VP test and natural reg are the same thing. Natural reg and VP test. So that when you go to bacteria, you are going to study about the chemical. We will be talking about VP test positive, VP test negative, MR negative, and so on, so that you'll be able to understand what we are talking about. Oh, someone is writing message. Hey, someone is not hearing.
We stopped hearing.
Are you sure? I'm there.
Yes.
Can you hear me now?
Yes. I don't know about
Okay.
others.
Yes, we can now.
Oh, I don't know what's...
Your voice is breaking up, so sometimes it's gone, sometimes it's up.
Oh, so sorry. So what's causing me this problem? I don't know. Because But my internet is very stable. I don't know. So yeah, you just you let me know if it continues, then I will have to present with this, and maybe I use another device to talk. Probably if it is my computer bringing problems.
Maybe
Then I
you
will,
are microphone.
no, I'm not using microphone. So I'm just using like computer, only computer.
Yeah.
Because I didn't want to connect microphones, because when I connect them, I cannot hear you. So I'm just using my laptop so that I can be able to hear you. But if not, if it breaks, then I will have to change to my I will have to use my phone or something else around.
It's
Just
now
to,
clear.
okay, if it is okay, then we are talking about principles here. So we just what I want from you is try to understand this test, because it will be very easy for you to understand, to remember, like for example, when you want to remember this, to remember this is used for this. So it is important to understand the test now, first of all. So that's what Mr. Teza was telling us. Biochemicals are confusing, but actually they are not. They are not. So we just first of all want to understand this biochemical test now. What do they stand for, and why do we use them for bacteria? So that's what we want to know. Okay. Now, what is the ONPG simply, and why do we use it? Hmm. Uh, Tessa, are you speaking? Because your microphone is on, or someone else can tell us, what do you know about ONPG test? ONPG?
ONPG, there are those organisms that address lactose fermenters that they delay, so we can use this to confirm.
Yeah, they delay. So it means those bacteria delay. So they are called delayed lactose fermenters. So why do they delay? In fermentation of lactose, we have two major enzymes. Two major enzymes that a bacteria should possess in order to ferment lactose. First of all, we have this one. Can you see this one? Beta galactosidase permease, and then we have beta galactosidase. So these two bacteria, two enzymes are different. And a gram-negative bacteria, which is able to ferment lactose, must possess them. First of all, this one is called beta-galactosidase permease. This is an enzyme that helps a bacteria to absorb, to capture a lactose, to capture the lactose. So they're known as beta-galactose permease. So the second enzyme that is important is this one, beta-galactosidase. This is the one that breaks down lactose. So which means a bacteria must possess both in order to capture lactose and to break it down. This one, permease, is for capturing. This one is for breaking down. They are permeable to galactose. I don't know. I don't actually. Okay, galactose permease. That's like to absorb, and then galactosidase is to hydrolyze. So in this case, when a bacteria doesn't possess, for example, this permease, but it possesses only galactosidase, it means capturing of lactose will be very slow to take up some amount of lactose, will be very slow. But still, it will take some time, but still a bacteria will be able to capture it, even without this enzyme. That's what it means. So a bacteria can only possess this. So that's why in 24 hours, if a bacteria lacks this enzyme, in 24 hours, lactose fermentation is negative because the bacteria has not yet taken enough lactose to break down. That's what it means. So then, after 48 hours, bacteria has taken enough and it will use this enzyme to break down that lactose. And of course, after 48 hours, lactose fermentation is positive. Before 24 hours, it was negative because of that reason. Okay. Now, but in order to to avoid wasting our time, we have another molecule that can be broken down by this enzyme, beta galactosidase, because it is the only enzyme that these bacteria possess. It is beta galactosidase. So that substrate is called ONPG substrate. So we don't want to waste our time. We just put ONPG substrate so that a bacteria can break down this ONPG and then from colorless to yellow color. And then we confirm immediately without waiting. So which sample do we use for ONPG? Is a sample that we tested for lactose fermenters for 24 hours, but it is negative. Because we don't want to wait for 48 hours. So we just want to cut it short. We say, okay, if it is negative at 24 hours, let's put ONPG. If it breaks down, we call it delayed. If it doesn't, we call it non-fermenter. So that's the purpose of this test. I think that one is a bit clear. So it means now, so we have some bacteria, few, but are not many. That we have a few bacteria that are ONPG positive. So if a bacteria is ONPG positive, don't look at lactose fermentation again. That must be negative. It means lactose fermenters negative because we test for, we have to confirm at 24 hours. ONPG positive, it means lactose was to be fermented in 48 hours. So that's what makes a difference. So there's a difference between lactose fermentation and ONPG positive. So these tests are opposite. Okay, so that's it. So that's what we have to do for this test. If you're okay. Okay, can someone tell us about Vox, Polska test, and meta-red test? These are the similar. It's also almost similar. We usually interpret them together. And you tell me why a bacteria cannot be positive for both of them. Then we continue with other things. Let's try to share. Someone can tell us. So what do you know about the two tests? This one is okay. I'm waiting.
So I think the homework is about
Yeah, we are doing
glucose
brainstorming.
metabolization.
Okay.
But in both there is glucose metabolization, but the stage at which
Glucose
or the amount of acid that are being produced differs. In MR, you have in MR you have up to pyruvic acid, but in VIP The pyruvic acid which is produced is further converted into acetone. Acetoin.
Acetoin.
Yes.
Mm, okay. Uh, yes, almost a big part of it. Um,
This
George.
is how I understand this for the sake of MCQs.
Yes, no problem. Yeah, for the sake of MCQ, that's okay. That's very good. It's very good level. You can still make it even if you have a very tough MCQ. George, we have not heard from you.
And then,
Um, George,
Yeah,
George, how are you?
I'm good. How are you?
I'm fine. So, not yet heard from you now. This time, we want to hear from you.
So Steve, Vogas Proscan methylurate, right?
Yes, yes. Why do we use them? Why? Why do you want to employ them in our testing?
I think I've forgotten about it a little bit.
It's all right. We are going to make it clear, very clear.
Okay, thank you.
Someone was on to talk about it. Think of it.
yes. The thing I wasn't going to talk about, VP test is used to detect a bacteria produce acetone during glucose fermentation. So that's all I can say on that. But when it comes MR test is kind of to the bacteria detect to detect the bacteria detect uh, acid fermentation of glucose, which maybe can produce can produce stable acid in production.
Okay, very good. Now, just one question so that I can also try uh to put some light on it, and then we continue. Why do we say that a bacteria can either be MR positive and VP negative or vice versa, but the bacteria cannot be positive for both?
I think because of the pathways that they take during the fermentation.
Which pathway? Because both of them take the same pathway. Acid. We are talking about acid production of glucose fermentation. Why one of them does not, like a bacteria cannot be VP positive and MR positive at the same time.
I think because they choose one pathway. I don't know.
Okay. Now, okay, let me try to clarify this one, and also I will also talk about why do we employ this test? Because we have many other tests you can use, like OF test. OF test also shows us the glucose something. But why do we also bring this test, MR test, and this one and the VP test? So the reason is we want to test a bacteria which ferment glucose to produce a mixed acid. It is called mixed acid pathway. Mixed acid pathway. So there are many acids that can be produced. For example, when you talk of mixed acid pathway, that's a bacteria that is able to ferment glucose and produce pyruvate, acetic acid, and lactic acid, for example, and many other acids. There are many other acids that can be produced from glucose. So that's what we call mixed acid fermentation pathway. So we use MR test or methylurate test. So, but why we don't need to use it if a bacteria is lactose fermenter? We don't need that because we only use it if a bacteria is only glucose fermenter. That's when you can employ MR because there's no need of testing MR for Escherichia coli. MR doesn't need that. We don't need that one. So even if we can say, okay, let's say it is positive or negative for this or this, but there's no need of testing MR test for Escherichia coli, obviously, because we use it for bacteria that are not lactose fermenters, but they are glucose fermenters, but they can ferment glucose in different pathways. I mean, the same pathway, but producing different types of acidics or acids. Now, but we are trying to say there is a reason why a bacteria can do MR test positive, and I'm going to bring a table. I summarize all, and I'm going to share with you after I finish this. I summarize the table showing all biochemical reaction of each bacteria we have in our microbiology. If every bacteria with its chemical reaction, I summarize, so I'm going to share with you, and you will find that wherever we talk about MR test, bacteria can be positive for MR test, but negative for VP test, because methyl red is a production of acid, whether it is one type of acid or a mixture of acid, but bacteria will stop there. That's what we call MR test. So it means that a bacteria is not able to continue breaking down those acids. That is what we call MR test. So bacteria can ferment glucose, yes, but it will not proceed. It will just stop there after acids are produced. So, but if a bacteria is having a capacity to be VP positive, it means those acids, they will also be broken down. And for example, pyruvate can be broken down into acetoin as another metabolite. And what we are going to test, it doesn't mean that the acetoin is a last product. It can also continue to break acetoin down and make it something else. But don't care. So what you are caring about is production of acetoin, which can be detectable in VP test. We detect it using alkaline molecules like sodium hydroxide or potassium hydroxide. And then after that, we can say that this bacteria is VP test positive because those acids produced in mixed acid pathway, they are broken down further to produce acetoin. It means VP test positive. But if a bacteria was not able to break them further, it will stop there and it will say bacteria is MR positive, but VP negative. So that's why we say that a bacteria can't be. Is it here? I think it is here. Let me see if it is written here. Oh. What is it? This one is what? Okay. A bacteria is usually MR positive or VP positive, not both, though some may be negative for both. So which means, yes, that is true because some bacteria are negative for both. For example, what did we say? Pseudomonas aeruginosa, it can be negative because Pseudomonas is a non-fermenter. So it can be negative for both. But some bacteria that ferment, like Proteus, Salmonella, Shigella, Edwardsiella, Providentia, all of them, Citrobacter, all of them can be positive for each of these, either VP or MR. Okay? Yeah, the other things are just like carbohydrate. This one is what? Amino acid utilization. There's something that you have to remember. This one. Okay. Amino acid utilization is just like to break down the amino acids and produce, especially to remove carboxyl group for amino acids. And the amino acid we use usually in amino acid utilization test is lysine or ornithine, and you produce something. So now, someone can also talk about it a bit. Yes, this is a utilization of amino acids, decarboxylation of amino acids or dehydrogenase. But decarboxylation is okay because that's what usually appear in our example. Mm-hmm. Mm. Okay, tell us about this. Decarboxylation, amino acid utilization. So, what about it? And what are the products of that? Mm-hmm. Mm. The carboxylation of amino acids. So decarboxylation simply means removal of carboxylic acid, and then there is a remaining part. There's a remaining of part of amino acid, which is now amine, or diamine. What do we call diamine? So diamine is like amino acid. We have amino acid. It has amino group and then carboxylic group on the other end. But between carboxylic group and amino group, there is a carbon chain. CH3, CH2, CH2, CH something. Then you have a CCO2, COOH. And then we have also on the other side, we have amine group. So what happens now, a bacteria can break down this, can remove the carboxyl group in form of CO2. It makes, because it is a COOH. It just remove it and make it CO2. And what is remaining there is called diamine. Diamine is the remaining part of amino acid. And instead of becoming alkaline, instead of keeping the acidity of amino acid, now it becomes alkaline. And then there's a production of this color. You can see it become purple. It was acidic. It was acidic before. It was yellow. After deamination, decarboxylation, it now become purple because of CO2 produced. It makes it purple. Purple means now alkaline shift. Okay, because of amine produced and CO2 produced in the medium. So we use also we use the we use this but we use this culture media that contains amino acids like ornithine amino acid and lysine amino acid. Especially we use Miller carboxylase medium. So this one contains those amino acids. So molar carboxylase medium, which contains sugars and then contains amino acids. So you'd see this one as you read. So the same as citrates. Citrates is also, especially when the bacteria is able to grow in what we call Simon citrate medium. So, and also diamination is just like to remove amino group. So we call it, in most cases, we use a phenylalanine deamination. So bacteria can be phenylalanine deaminase. So, for example, in a case of Enterobacteriaceae, Proteus, Morganella, and Providentia, they are diaminase positive. They remove diamine. They remove amine group from phenylalanine, which is also amino acid. Then we obtain pyruvic acid, which is produced. Pyruvic acid, and the color would change from yellow to green. The green color would be produced because of indicator iron chloride. So it will become green. So the same as DNase. DNase is very simple, isn't it? DNase is very simple. It's just like medium contain DNA, and bacteria can produce enzyme called DNA enzyme, which break down DNA. And if DNA is break down or hydrolyzed, it usually doesn't precipitate after adding hydrochloric acid. It doesn't precipitate. So that's what makes it. That's what makes it still like this. If it precipitates after adding hydrochloric acid, the medium will keep this color, green color. But if it is cleared around the colony, there's no precipitation because it was hydrolyzed by DNase enzyme. So it will look like this. So then you could say that around this medium, around this colony, there's no precipitation of DNA, then it becomes like this. Okay, so all of these tests are indole production, you see them. So what is that done here is production of indole from tryptophan, and then we use a COVAX or enrich reagent. This test in order to extract and to detect indole produced from tryptophan amino acid. So this oxidase is what I say we use for all Enterobacteriaceae. All of them are oxidase negative, except Pseudomonas shigelloides. Pseudomonas, all of them positive. Neisseria positive, Vibrio positive. So, which means, let's give you something here. The rest of gram-negative bacteria, most of them are oxidase positive, except Enterobacteriaceae. And I don't know if you can recall all bacteria that we have in Enterobacteriaceae. Okay, can someone tell us what bacteria do we have? We are talking about Enterobacteriaceae, but I'm curious if you can recall those bacteria you're talking about.
Yeah, we're talking of E. coli, Klebsiella, Citrobacter, Enterobacter, like the Morganella, Proteus providencia, Salmonella yersinia, that kind of, yeah.
Oh yeah, yes, all of them you have already listed them. But how do you classify them? We have two classes
I think
of
I think we classify them according to lactose fermenters that produce pink colonies, then non-lactose fermenters that are colorless.
We also classify them according to their pathogenicity. Some of them are not pathogenic, others are. For example, the pathogenic one, we have only 3. And yes, and that is the best way to classify them in order to avoid confusion. Because if you classify them as lactose fermenter and non-lactose fermenter, yes, it is also good and it is the best approach, but knowing them as pathogenic, non-pathogenic is very helpful. For example, you know that pathogenic ones we have, or there are only 3. We have
I think of the Salmonella, Shigella, Klebsiella. I don't know.
Salmonella, Shigella,
Yersinia.
Yersinia species. So Yersinia species, Yersinia trichotica is very bad.
Mm-hmm.
Yersinia trichotica. So Yersinia, they are there. So we have Yersinia, we have Shigella and Salmonella. So which means anytime you find them in sample, they are all pathogenic. Anytime, so you cannot say that this is that stool, or they are always pathogenic. But when you talk of Escherichia coli, we also have Escherichia coli that is pathogenic. For example, enterohemorrhagic, very pathogenic. But still, we classify it as opportunistic pathogen. But what you call Escherichia coli 015-7H7, that is very pathogenic. But generally, Escherichia coli is not pathogenic. It can be found in stool. For example, if you find Escherichia coli in stool, are you going to make it a big deal and report it? No, you can't. Because Escherichia coli must be in stool. That is normal. It is normal in stool. It can be abnormal when that when that patient is complaining with symptoms. Maybe you can think of what, maybe hemorrhagic, enterohemorrhagic maybe with the stool with the blood or enteropathogenic. So some symptoms like diarrhea, yeah, but normally the nature of it is not pathogenic. So that's what I'm trying to mention when when it comes to enterobacterial cell, which means all of them are oxidase negative. And Pseudomonas, Neisseria, all of them positive. Not only this, we have also a group, okay, Neisseria species, all of them, and even Moraxella, and Haemophilus, they're also positive for oxidase. We have also Vibrio cordeli, Vibrio species, they're also positive. Campylobacter species, they're positive. Ailomonas species, they're positive. Presiomonas species, they're also positive. So, which means the oxidase is useful and it can highlight Enterobacteriaceae, say all of them are negative. The rest of gram-negative are positive. Yeah, you can say that. Maybe we could find some little exceptions, but not many. I can assure you that most bacteria we find in gram-negative, except that group of Enterobacteriaceae, all of them are oxidase positive. And oxidase is very useful tool to test. Okay. Now, so COVAX is what to use. You remember, you can use COVAX or can use Enrich, but that's a little difference is that there's what you call extraction. When we use Enrich, we have to add xylene for extraction to extract that. Oh, sorry. I was trying to mix up. I was mixing up things because I'll talk about indole and then oxidase. So because COVAX is also used. So, but the reagent for oxidase is called COVAX oxidase reagent, simply like that. So, but we have also COVAX reagent we use for indole production. So it's called PDA. It's called it is a long term. So I don't want to repeat it. Sometimes even if I'm reading it, it disturb me so much. So, but it is called PDAB. So it's called COVAX reagents. You can use it. And if it involves extraction of indole, you have to use xylene, and that's when you employ enrich reagent. So, and then the color will be produced, and you confirm that bacteria is indole positive. For example, like Escherichia coli, which is indole positive. The same as urease and motility test. So, motility test, bacteria can just move motile. For example, which bacteria is motile? Bacteria must contain pili, and then pili or flagella. So all of them can be motile. So especially those ones that contain H what? H antigen. H antigen. We have H antigen, O antigen, and K antigen. Somatic, capsular, and flagella antigens. So if it contains H antigen, it means bacteria will be able to move and it will migrate. And we find when you introduce, because we use a semi-solid medium, we don't use a liquid so that we can block them. Those ones that are not able to move, we block them. But those ones that are able to move, they will have to move across this medium. So that is it. Now, I think this test now at least now you understand that the use of the test, because the test we use for gram positive are not difficult to understand. You know, we have first of all hemolysis, beta hemolysis, alpha hemolysis. Those are the tests we use for gram positive bacteria. We have others like CAMP tests, CAMP androlysis test, biostracin, optochin, all of them, biosolubility tests. Those ones are very simple, and we will see them when we are trying to deal with the gram-negative bacteria, gram-positive bacteria, but they're not really very, very difficult. So now, someone was saying that before we go to mycobacteria, someone is saying that using these tests can be challenging. How? So how? So I think for me, the way I can help you after understanding this test, maybe I can give you a table. Yeah, so challenging. Yes, I know. maybe I send you a table, each bacteria with its own reaction. When when it when you like for example, when you have okay, it takes time. Yes, I know, and I can assure you that you can't go for exam with the full microbiology in your brain. It's not easy. It's not easy because even I know I did exam, so I did exam, and I was not like the time I went for exam. Oh, I was not. I was like going back to read BOC questions, and the time I did my exam, we only have edition 5. It's 5. I think it is. Yes. You know what? Now we have edition 7. We have edition 5, edition 6, edition 7 of questions. The time I did my exam, only what was out was edition 5. And edition 5, questions in edition 5 they're not so difficult. So when you look at the edition 7, they're more complicated. So even me, I was like reading my questions outside when I was going to the exam. I was sitting outside and read questions. Oh, I was like, oh my God, how am I going to make it through? Yeah, but still, uh, we don't need to have to make it 100%. At least to get pass mark is very essential. Someone is saying, especially the fact that if one is not working in microbiology lab, yeah, that's true. I think someone working in microbiology lab, this is very simple deal. But not all of us. Even me, I'm not working in microbiology lab and didn't work there. Except like having to work in microbiology lab when I was at university, like maybe doing something there. But not all of us, not all of us. But it's okay. We can just read and understand.
But I feel like even if you're working in the microbiology lab, Africa, with most of the biochemicals, we don't even use them.
Yeah. That's true. Yeah. So which hospital that is using all of this?
Ah,
There's no way. And of course, there's no way we can use all of this. So for me, I don't even understand America, how they really want us to know all of these things if they really use them. I don't know, maybe if they use them. And there's a problem that Mutesi is pointing out, which is automation. Automation is everywhere. Even sometimes you find in lab there's automation where you don't even culture and read. The machine will read for you and tell you immediately what is there, without even needing to read and try to know what is this, what is this. But when it comes to exam, it seems like you have to know everything we have to work to replace the automation in exam. But when it is at workplace, automation will replace us. So, all right. So I'm going to share with you that table. It will help you, I know, because I summarized it. I prepared it very carefully. And I think it is going to help you to try to classify them, to put them uh, in your own organization. You're going to organize it accordingly. So now, apart from this, let's maybe talk a bit about mycobacteria, and I will try to also show you the secret in mycobacteriology. TB. Okay, let me try to move this one. Okay, just allow me to grab my laptop charger so that it doesn't get off. It's not near me. I have to pick it from somewhere else. Let me pick it, and just 1 minute I come back and we proceed. Okay. Sorry, I'm back. So let's try to think about mycobacteriology. And so how can we study? How can we read mycobacteriology? Okay, mycobacteriology, as you have seen, what we are offering as microbacteriology is like what we are offering is very vast and very wide in in what is done in lab, especially what is done in lab, because we cannot do all those things in lab. So we have so many microbacteria there, and the thing is that, and the challenge is that even if they are not present in our labs, but we are using them. We are using them in exam. So we need to know them. We need to understand them. So now, in order to make sure that we classify, microbacteria is not really challenging, because microbacteria, when it comes to biochemical, when it comes to media that are used, there are very few. It's not like general bacteriology. But microbacteria are also many, but also they have very narrow differences in terms of their chemical reactions. There's so narrow differences, even in their growth. In their growth, some of them are known as slow growing. Others are known as fast growers or rapid growers. Others are known as, of course, slow growers. Yes, you can say that some of them grow in 3 days. Others can grow in 2 weeks or 3 weeks or 6 weeks. So those are what we call slow growers. And slow growers are many, many, very many, because even most pathogenic are slow growers, like Mycobacterium tuberculosis is a very slow grower, mycobacteria, which can take even up to 6 weeks to grow. And it just uses one specific medium, which is called Lowenstein-Jensen medium, which is actually what is used in most labs. Lowenstein-Jensen media. But also we have other media that can be used for mycobacteria, for example, tuberculosis. For example, what we call Middlebrook 7H10. So Middlebrook 7H10 is a solid medium that is used, and it is made in albumin, not egg. So that's the difference between egg-based and albumin-based medium. Lowenstein-Jensen medium is egg-based, but Middlebrook 7H10 is not egg-based, is albumin-based. So you find also albumin, you found potato flour, and salt. Okay, there's some contents of that. So we have also what you call Middlebrook 7H11 and Middlebrook 7H9. Middlebrook 7H9 is liquid medium, it's not solid. So it's liquid medium, but albumin-based. Whereas Middlebrook 7H10, H11, it is still also a solid medium, but made in albumin as well. So that is what you will find in the exam. Mostly they will be asking you classify or try to classify alkaline-based, albumin-based, egg-based, from solid and liquid. Each of them, liquid and solid, selective, non-selective. So when, for example, when you want to like selective media, you can take Middlebrook 7H10 and you add some bacteria, some antimicrobial, for example, like Proximi B, vancomycin, they can be added, and you make them just selective. Other culture media we use there in mycobacteria can be like Petragnani and American Thoracic Association medium and Petragnani. Only those medium are used in mycobacteriology. So you can see there are very few. You can memorize them. I don't need to memorize them because, yeah, you can memorize them maybe in case of you find that they are asking questions like to classify them in their groups, but they're not many for sure. So now is that we can classify mycobacteria like two major classes. We can classify it as MTB, as tuberculous mycobacteria, and then classify them as non-tuberculous. So non-tuberculous and then tuberculous. So tuberculous are those ones that cause tuberculosis. For example, we have Mycobacterium tuberculosis, but also we have Mycobacterium bovis, for example. Where is it? Bovis. These are all of them are tuberculous. Tuberculous, we have tuberculosis, bovis, africanum, carneti, microti. All of them, they are what we call tuberculous mycobacteria. So which cause tuberculosis? They're all cause tuberculosis, similar tuberculosis. If you have M. bovis, Mycobacterium bovis, it is not MTB, but it still causes tuberculosis. The same as africanum, it causes TB cases. TB cases in tropical Africa, like my country is in tropical Africa. So this one it can cause TB cases. Maybe sometimes we are dealing with africanum here as the cause of tuberculosis, not exactly the TB. So then, TB pathology, we cannot go through it. It's like respiratory. Then after respiratory, we have what we call pulmonary TB, but it can extend to blood, which is called miliary TB, or can reach other organs through blood, which is called miliary. Then we have also reactivation TB, which can be reactivated in case immune system declines after even like sometimes remission or absence of symptoms, and then it reactivate again. It can happen because of low immunity, especially cellular immunity. And then you have what we call extrapulmonary, which is known as also miliary TB. So extrapulmonary. It is also similar to miliary TB because it can spread through blood, then it reaches other organs. So what makes every sample possible for TB? You know, someone with TB, you can take a urine, you find TB. You can take swab, there's a TB. You can take blood, there is TB. You can take stool, there's TB. Sputum, there's TB. So in most cases, because most organs are affected, when it is extrapulmonary, any sample can give you TB that you want to see. So that is about mycobacteria. And how do we identify them? Now, we have identification, the biochemical test you are talking about. Let's look at Mycobacterium tuberculosis. MTB, there are major tests that you want to know here. First of all is niacin. Secondly is catarase. Then you have T2H. Then you have NAP. Okay? So that you can identify MTB or Mycobacterium tuberculosis. And you know, in most questions for microbiology, they are usually looking for MTB. So they can also ask you others like avium complex, Mycobacterium avium complex, or MAC, but this is the most common, MTB, Mycobacterium tuberculosis. So first of all, MTB is a niacin producer. Niacin test. Niacin is called niacin accumulation. So why do we call niacin accumulation? Even though we are not looking at the test how it is done, but niacin is a product that is produced by most of Mycobacteria species. But niacin, we have some other bacteria apart from MTB. It has niacin hydrolyzing enzymes. So which hydrolyze niacin and make it or convert it to another metabolite. But MTB doesn't have capacity to metabolize niacin, but it will accumulate it. So that's why we call it niacin positive, or we can call it niacin accumulation positive. So MTB is niacin accumulation positive. And then secondly, it reduces nitrate to nitrite. This is very common for most mycobacteria. It is not specific for MTB, but niacin is a bit specific. Though we have some few other mycobacteria, but which are not Mycobacteria tuberculosis. I mean, which are not tuberculous mycobacteria, but they also accumulate niacin. I will show them. For instance, them like Malimoensis. Malimoensis also is niacin accumulator. But I'm also going to show you another thing that can help you to differentiate between this and any other niacin accumulator. For example, it is not written here, but we'll see it somewhere else about pigment production. Mycobacteria species, we are also concerned about pigment production. So you have to remember that MTB itself doesn't produce any pigment. For example, we have this is a Lowenstein-Jensen medium. You can see Lowenstein-Jensen medium is buffy in color, isn't it? Buffy. So the color is like this. So whether you expose it on light, whether you don't expose it under light, it is buffy color. So that's what makes it non-chromogenic. So when you expose to light, there's no color production, there's no color. Usually color is yellow or orange. When it remains buffy, it means it is a non-chromogenic. So MTB is non-chromogenic, but one or two mycobacteria species which are niacin positive, both of them are chromogenic. So that will help you. Always keep in mind MTB is a non-chromogenic and niacin producer. These are the major characteristics that I'm trying to show. Secondly, you may be confused sometimes if they bring bovis, Mycobacterium bovis, and maybe Africanum against MTB. You can be confused. But remember, we have something that's called T2H. That's what I also mentioned. T2H is very important. T2H is called tyrphene 2-carboxylic acid hydrazide. So this T2H it helps you to differentiate between bovis and MTB because both of them are non-chromogenic. You can imagine if you are given this one, how can you know it is MTB? Because bovis will also grow like this. If you grow a bovis, it also grows like this. So for you to know, expose both of them to light. Like this one exposed to light, it can also give you conclusive result. So because both of them are non-chromogenic, so it is still confusing. Now use this one, T2H. T2H differentiate between M. bovis and MTB. M. bovis is inhibited, but MTB is not inhibited. So it means MTB can grow in the presence of T2H, whereas M. bovis cannot grow. MTB is niacin accumulator. In fact, M. bovis is not. That's also something that is good. Good news. MTB is niacin accumulator. M. bovis is not accumulator of niacin. So, but also other things can be similar. So that's what makes it different. So remember, MTB doesn't produce like catarase enzyme. It doesn't. When we say catarase enzyme, it must be enzyme that is heat resistant. Heat resistant catarase enzyme. What is heat resistance? So what we do, we just put catarase, hydrogen peroxide, because it's a substrate for catarase. After that, then we do what? We do warming. We just warm them or heating them. We heat them to 60 degrees. When it reaches 60 degrees, catarase disappear. That's why MTB is called heat stable catarase negative. So it means it produces heat labile catarase. So it doesn't produce heat stable catarase, but a group of isoniazid resistant, they don't produce any catarase. So it means these ones that are known as isoniazid resistant, that is what we call what is not extended spectrum. That is what we call multi-drug resistance. Multi-drug resistance, which is isoniazid, because isoniazid is the first line, so they don't produce any catarase. So those are the things that you can use for mycobacteria. First of all, non-chromogenic, niacin accumulator, growth in the presence of 2,2-H, heat label catalase production. And so when you look at all of these things, can help you to distinguish it from others that may look similar, like bovis, like others like malmoense, which can be niacin accumulator, but it does not exhibit other characteristics like this. Okay, someone is saying network is disturbing. Sorry, maybe try to boot your network. I don't know what happens. So, okay, are you okay with this with this explanation here? Are you okay? Are you okay? Or have something? You have something to add? Tell me, or tell us. Mm-hmm. Any any um any suggestion here on this on this? Yeah, this is MTB, and then it looks like this. So treatment is what we have to read and understand to understand. And then when it is not when it is resistant to the first generation of drug, we have to withdraw drug and use the second generation of drug, which include also fluoroquinolones and aminoglucosides, because already isoniazid and rifampin which are the first line of drug, there is resistance. So we use the second line of drug for extended periods. So, but if this also there is a resistance against aminoglucosides and fluoroquinolones, then there's what we call extended uh, extended or extended drug resistance. So it means it is resisting against first line of treatment plus second line. Second line is fluoroquinolones. And also aminoglucosides. So when this happens, extended drug resistance, so it means we are going to do susceptibility rapidly because there's no other drug remaining. So we can just do susceptibility so that at least we can find one or two drugs that can be used, because line of treatment for TB are only two line of treatment. We have first line and second line. So, but if also second line there's resistance, so we have to do susceptibility testing at least to identify any remaining drug that can be used for the therapy. But otherwise, this one is very difficult condition. So I talked about bovis. Bovis niacin negative, nitrate negative, and then don't grow in T2H. Very distinguishing from MTB. You can see Mycobacterium bovis, of course, the only thing can be used to distinguish it from MTB is niacin negative, nitrate negative, and the absence. There's no growth in T2H chemical. Okay, so we have then what you call NTM, non-tuberculous mycobacteria. So this one is where we find the slow growers, very, very, very pathogenic, Mycobacterium avium complex. So what is unique for this, what you call an non-TM? Non-TM, what is unique is that they don't cause any TB, but they cause similar respiratory syndromes and cutaneous skin problems and respiratory problems, but not TB. So NTM can be Mycobacterium avium complex or MAC, and also intracellular because they are together in a group, so we don't need to distinguish them. So they are reported as MAC complex, and so they cause, of course, problems like respiratory problems. They cannot even disseminate in other organs. But the big thing is that treatment for them, we can use just excisional surgery to remove the nodules because in case they're cutaneous, but if they're not cutaneous, just use other drugs like the drug we use for TB. They can be used like isoniazid, they can also treat them, and also tambutol and fluoroquinolones if it is required. Tambutol, rifampin, chlorphazamine, and even amglucoside, they can be used. So diagnosis of MAC is very easy. Let's see what you can use to identify them. Heat stable catalyst production, MAC complex. Heat stable catalyst production plus growth in 2,2-H. So which means that MAC complex is similar to MTB when it comes to T2H, but heat stable catalyst production makes it a difference. Heat stable catalyst production, because for MTB, it is heat labeled catalyst production. Otherwise, every other thing is similar, which also brings to attention that there is non-pigmentation. So which means this MAC complex also, there is no production of pigments upon exposure to light or without light. There's no pigment production. The same as Kansasai also. So Kansasai, this one is also an anti-M, an antituberculous. Yeah, but this one also, the good thing is that the treatment of this one is like others, like nifapine and ethambutol. But let's see how we can identify it. Kansasai is actually identified as a photochromogenic, so which means that Kansasai that produces pigments when you expose them to light. They produces pigments, and some of them can be scotochromogenic. Scotochromogenic are those ones that produce pigments either in light or without light. So like this one. So you can see this one is light production. We use Lowenstein-Jensen. You know, Lowenstein-Jensen medium is very commonly used in lab. Maybe it's available everywhere, because even so where I worked in my lab, we had this one, Lowenstein-Jensen medium only. But you could use it for everything. It is egg-based medium. So when you expose Kansasai to light, it is photochromogenic. Without light, there's no pigment production. So then it is catalyst positive, but key features are here. MTB like growth, right? Photochromogenicity makes it a difference. Photochromogenicity because MTB doesn't. Twin 80 rises in 3 days. So we don't use this test for MTB actually, but nitrate we use it. Similar to MTB. Catarrhase similar, but this one produces a heat stable catarrhase. MTB doesn't. And the plasminogen production. And you know, MTB does not produce plasminoids because it grows it grows in plasmamides. So not like bovis. So these are some of the characteristics. So you can see the difference is just color production and twin 80 rise. So what you just have to know, maybe Kansasai is the one that we talk about twin 80, and you will find it in questions. If they bring twin 80, you have to know. So Genafis, we have others like Genafis. So there are not so many biochemical tests you can use to identify this one, but it is very hard to grow. Usually we can use a backtech system. Backtech system is very automated system, very tough system. We use to grow those ones that are very fastidious. It doesn't grow easily, Genavens. But even if we use middle block 7H11, we have to supplement it with mycobactin. Mycobactin is a growth enhancer. So which means that Genavens is very hard to grow. Yeah, so, but it produces heat stable catarrhase, and the plasminoamidase and urease test are also positive for this one. Plasminoamidase enzyme is also produced by Genavens. So hemophilum is like for hemophilum is a bit different because hemophilum can still grow on chocolate agar. Not only mycobacteria culture media, but hemophilum can also grow on chocolate agar and Hillermington agar. So you can imagine it can grow on bacteria media. So like chocolate agar and Hillermington agar. Because it is a morphilam, it depends on X factor. X factor is the hemine factor. So hemine is found in hemoglobin, and hemine is found on chocolate agar. And sometimes when you add some blood on Millington agar, it can also contain hemine, and you can use it to grow hemophilam. So hemophilam can grow also, of course, on other microbacteriology media like Middlebrook 7H10, but you have to supplement it with the hemine, first of all. Maybe you can place a disc, it grows around the disc, or we can mix it with the hemine, so it supports growth of hemophilam. So that's how they look like, non-pigmented, and very simple, non-pigmented, it is not pigmented. And also, of course, how you know is that they would also bring these things. They will bring chocolate, they will bring blood, hemoglobin, hemine. You know that if you're dealing with mycobacteria, it is now hemophilam, okay? That is it. Malimoense and so on. So we have these others. We have these others also. So not so common. Malinum. So the not so common. So the key point here is to try to list down some distinguishing features. That's the key. And I'm going to share with you the table. I have put all the features of each of these bacteria. So you will use the distinguishing feature at least to have idea about each bacteria that is here. Apart from this, so we have Simiei, we have Sclerotigae, which is very specific. Sclerotigae is very, very specific because this guy can grow, can grow in just only absence of sodium chloride. If there's no salt, it can grow very well, but in the presence of salt, it doesn't grow. Acylans, it is there. Xenopi, Xenopi is unique because of temperature. It can grow at 42 degrees. So when you sit for 42 degrees, you know it is a Xenopi. Xenopi is the only mycobacteria that grows at 42 degrees, and it grows very well. But when it is at 37 degrees, it doesn't grow. Whereas most other mycobacteria grow at 37 degrees. So rapid growers, we have some groups like these ones. We have Abscessus, Coronae, Fortitium. Those grow very rapidly, and these are key features. So R-cyphatase is an enzyme. Nitrate actually negative growth in McConkey agar without cholesterol. This can be actually very, very good indicator or very good feature of this, what we call bacterium, Mycobacterium abscessus group, and then Colnay group. So, and then we have also Fortitum segmatis. These are all rapid growers, and then we have the prey, which does not grow in laboratory culture media. This one, the plant doesn't grow at all in lab culture media. It does not grow. So identification of this one is just to count after staining. We have to stain, and then we count them according to what we call bacteriologic index, and then what we call morphologic index. So bacteriologic index, it is to count how many staining bacteria are filled, but morphologic index, we have to count 100%, 100 cells, and you have to know how many of them stain solid. It means the entire cell is staining solid. That's what we call morphologic index, and then we report it like that. So, I want to take you through a bit in diagnosis, lab diagnosis. Oh, am I still presenting? I want to wait for I'm not presenting. I think I'm not. Yes. This lab diagnosis for tuberculosis. Tuberculosis lab diagnosis. Lab diagnosis. This one. Okay. Okay. So lab diagnosis, when it comes to sampling is every sample. Any sample can give you something good. Respiratory sample, sputum, gastric washings, aspirates. They can all of them give you a good specimen. Urine also for TB. Stool also for TB can be used. Other body fluid like CSF, ascitic fluid, many other fluids. All of these samples, mycobacterium can be isolated from any sample. Then you have the process of what we call the digestion and decontamination. Okay, someone can tell us about this one, digestion and decontamination of specimen. So what do we use to decontaminate, and what do we use to digest, and why do we digest and decontaminate these samples? The floor is open. Please, let's talk about this one. So digestion and decontamination, the reason why we have to take some time on it is because these things are also continue to return in questions. Then why do we digest and why do we decontaminate samples? Okay, maybe that one can tell us. What do you think? Mm. Uh-huh. So can tell us what's the process of the contamination and then digestion, and why do we either decontaminate or digest, or why we do not digest? I mean, why do we do not decontaminate but digest? So someone can tell us.
I think I can try.
Yes.
So the purpose of this digestions is to break down the thickness of a mucus in the specimen, where we use NARC. But when it comes the contamination is to kill or to reduce other bacteria or fungi or in the specimen.
Okay, that's good. That's correct, very correct. So why do we do decontamination? Some samples can be digested. Others cannot be digested. They can just be decontaminated only. Other samples, nothing is done. Either decontamination or digestion, nothing is done. So for example, if you have a sputum, first of all, you have to digest it because it contains proteinous. It's called proteinous mucus. Proteinous compounds, which makes it like very, very mucoid. So like your sputum. Sputum can be digested, then decontaminated. So that's why we are using NALC, N-acetyl L-cysteine. Where is it? Oh, yes. N-acetyl L-cysteine. This is just a mucolytic. It breaks down the mucus. And then we use the contaminating agents, this one, sodium hydroxide. Okay, so they work together. So which means we need to decontaminate, because the reason why is that mycobacteria species they can be isolated from many samples, but they are usually there along with other bacteria, So like sputum, you find so many other bacteria. For example, someone could have Pseudomonas aeruginosa, even if Pseudomonas aeruginosa is also a bacteria, is also a pathogenic bacteria, but it is not what we are looking for. We are looking for MTB, for example. So we have then to decontaminate it so that we can kill it, so that we avoid overgrowth. Yeah, but it is also a good thing that if someone is having cystic fibrosis, we can also try to look for Pseudomonas aeruginosa in sample using the normal microbiology procedures because it was also pathogenic. So, but of course, we have N-acetyl-L-cysteine plus sodium hydroxide. Both of them, one of them is the contaminating agent, sodium hydroxide. This one is what? It is only mucolytic agent or digesting agent to make it like a liquid, not some mucoids. We do it in microbiology, in microbacteriology, in TB diagnosis. Even before you run junior expert, you have to decontaminate it and digest. We use those liquids in small bottles. Put a sample and shake. Yeah, what you are doing is to digest, but also to decontaminate because both of them are there. L-cysteine, acetyl L-cysteine is there, and even sodium hydroxide is also there. So we are doing both things together. We can also use others. For example, the benzalkonium chloride and sodium phosphate. So these are actually the digesting molecules, and oxalic acid is also decontaminating. But if a sample is very, very contaminated, like with the Pseudomonas aeruginosa, or very contaminated with the other bacteria, so we use oxalic acid. So when you don't suspect it, we still use N-acetyl and sodium hydroxide together. So staining is acid fast. Sometimes we don't stain with acid fast, we stain with the fluorescent stain, rhodamine, oramine stains. So this one is mostly used, rhodamine. So I think we are no longer using acid fast in many labs, but oramine and rhodamine are still used. But the thing is that even if you don't use them so much, they will appear in the exam. Zen, Nelson, and Kinyun stains. So both of them are acid fast. So because we decolorize with the acid alcohol and they resist decolorization. So that's why we call them acid fast bacteria. So culture media. Let me see if I can classify it for you. Where is it? Okay. Let me see how I can classify it. Okay. Oh, good. Yeah, it is very classified. Oh, I think we have everything now. So culture media that are used for mycobacteria, because all of them are strictly aerobic. So we are not talking about anaerobic, facultative, or whatever thing. All of them are just aerobic, unless we want to supplement them. For example, Genovese can supplement. Even hemophilum, we can supplement it. Marinum can supplement. So there's some of them that are very fastidious that need to supplement other molecules. But normally we have their media, which are classified under egg-based, serum albumin-based, and liquid media, but non-selective and selective formulations. Okay. Let me see if I can. I want someone to remind us once for all. Egg-based. Example of egg-based. But who can give us example of an egg-based medium for mycobacteria? Egg-based medium. Egg-based medium?
Western Jensen media.
Okay, Lowenstein Jensen media is egg-based. What about serum albumin agar-based or albumin-based medium?
We have the Middlebrook 7H10.
Mm-hmm. and 11, okay? Then we have liquid. Which one is liquid medium?
Middlebrook 7H9.
Okay, that's liquid. Yes, that's true. And then, okay, let's look at some selective ones. Formulations, because not just that we have other media, but you have just formulations. We can have formulation of, for example, we have egg-based. For example, we have that lowest suggested media, but you have others like American Thoracic Society media. So they contain egg plus others like potato flour, glycerol, malachite green, which destroy gram-positive bacteria. And you have their selective formulations like graft low suggested medium and mycobacterial. These ones actually they are what you call egg-based medium, but selective because after adding mycobacteria, after adding antibiotics, we make them selective media. And then we have agar-based. Agar-based, we have talked about all Middlebrook. We have Middlebrook 10 and then Middlebrook 11. So they contain albumin, and we can add some antibiotics. For example, we can add proximib B, afotricin, carpenicillin, timoteprimo, so that we can make them selective. For example, what you call Mitson. Mitson selective media is Middlebrook 7H11, but after adding antibiotic, it is known as Mitson, Mitson selective media. So that's them. And then you have liquid media, especially Middlebrook 7H9 broth. It is used as liquid media. And then you have Dubos twin albumin. It is also used as liquid media. And then we have different formulations, of course, to make them selective. We have a group of selective. For example, Bactec 460TB. This is a system. This is automated systems, and also MGIT. Especially these MGIT is used very commonly in lab for rapid growing of mycobacteria. So it is called liquid automated system selective for mycobacteria. So other culture media, because we talked about chocolate agar, for example, for Mycobacterium haemophilum, also Mycobacterium marinum can also be supported by hemin presence. Yeah, so these are other formulations, like other media. Even you remember that even in some of them can even grow on cryptosvirate free McConkey agar. So those are some of the other formulations that can be used. So colony of identification of these mycobacteria is just about how they grow, how they look like. Colony morphology, growth rates, and temperature. Those are the things. Colony morphology. What do we talk about? For identification, colony morphology, what are we talking about? Colony morphology?
Colony morphology, we talk about the size, the shape.
Size and shape, and then pigmentation. Yeah, it is also there. It is also added to morphology. Yeah, it is added as pigmentation because some of them they grow, but they start turning to orange and yellow because of pigmentation. Some others don't actually produce any pigments. So that is it. And how they grow as rough, they may grow as smooth. So it depends. And then you have growth rates. Some of them, of course, as you classify them, some of them are rapid growers, others are slow growers. The slow growers are the ones that we call pathologic. So most of these rapid growers are not even pathologic. So then We have so growth rate is like that. Temperature is also very important. Remember that Mycobacterium xenopi is very good grower at 42 degrees. It doesn't grow very well at 37 degrees. Xenopi is different. Others may grow at lower temperature, but most of them grows actually at 37 degrees. Most of them. But only these are the exceptions like Xenopi and others like Marinum, Ocellanis, Hemophyllum, which grow best at 32 degrees. Others, the remaining, even MTB grow at 37 degrees. Photoreactivity, of course, is one of the morphologic feature. Photoreactivity is some of them can be photochromogenic, others scotochromogenic, others non-chromogenic. Yeah, but remember MTB is a non-chromogenic. Mad complex, Mycobacterium avium complex is a non-chromogenic. Bovis is non-chromogenic. Astralis is non-chromogenic. But because you want to differentiate, maybe let's say which one? Kansasai, okay? Kansasai from Mark, because they share a lot in common. Kansasai and Mark. So know that Kansasai is a photochromogenic, whereas Mark is non-chromogenic. Yes, so that is it. And you know that Mark can grow even in T2H like MTB. So those are some of the chemicals that they can share in common. So scotochromogenic, and you can have Goldenei, Scorsgai, Scrophilasium, and Xenopi. That particular Xenopi, remember, it grows at 42 degrees. It also grows scotochromogenically, which means pigments produced in light or in dark, like this one. So we have also biochemical. You remember the niacin. Niacin, we remember it. So if for niacin specifically for MTB, let's agree. Niacin specific for MTB. Forget about others that can produce niacin. They are not essential. Niacin, it is good to identify MTB. And you know, ACP questions are not so tough. So they not go beyond niacin. If they put something niacin positive, oh, you have to think about MTB. Maybe you can look at other biochemical reactions, but niacin for MTB is a key. Nitrate for many, not only MTB, but MTB is one of them, which is nitrate. So it means you check niacin, you check nitrate. If nitrate is positive and niacin, oh, that one is MTB for sure. And also maybe something else can be added as well in the chemical reactions when you are asked the question. So we have then catarrhs. You remember catarrhs? We talked about catarrhs in terms of heat stable and in terms of heat labile. Some of them can produce catarrhs, which is resistance to heat. Others, actually, they are very variable. They will be destroyed by heating. Okay, like for example, MTB does not produce heat stable. It is always heat labile. And remember, isoniazid resistance produced which catalyst? Can someone remind us which catalyst that isoniazid resistant strain produces? Is it heat stable? Is it heat labile? Which one?
I think
Heat
labor.
Isoniazid produce what? Isoniazid resistant. This one is also somewhere in the question.
I think heat.
I'll show you questions maybe next day, if it is tomorrow. There's no problem. I'll show you where you find it. Okay. Can someone try to tell us which kind of catalyst that isoniazid resistant strain produce? I think I mentioned it, right? I see Jude there. I see Leo. Leo has not yet talked to us. Leo, how are you doing? I think in your place it's very early. It's morning time, huh?
Wow.
Leo, how are you doing?
Heat labor.
Eat level. Someone else? Leo. I'm talking to Leo. Leo, how are you doing? Are you around? Or sometimes maybe you're using your earphones. You're not able to speak with us. Uh,
Can you repeat the question, please?
I'm saying now we're talking about this kind of isoniazid resistant strain. We are saying that MTB don't produce any. I mean, they produce heat stable catalysts, but isoniazid resistant produce which type of catalyst enzyme? Which type of catalyst that isoniazid produces? Isoniazid resistant strain.
Heat
Someone told
labor.
us that it is heat stable. So what do you think? Isoniazid. Bianca, were you going to say something about this? Which one that isoniazid resistant produce because others produce heat stable? Mm,
I'm not quite sure.
I'm not sure. Who is sure here? In our list, who is sure?
Please repeat the question. Let me get
I,
it nice. Repeat the question.
I think you have already answered it. Yeah, we are saying that Mycobacterium tuberculosis complex lack heat stable catalysts, but isoniazid resistant produce another type of catalyst. Which one?
Heat labor.
It love by, yeah.
And
He-
I
he-
wanted to misguide you, and you have followed me.
no, it's it's it for 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 isoniazid resistant, meaning they have heat. They produce heat stable catalyst.
And you are still following my misguidance.
Yeah, sure.
Yeah, I'm saying this because I've seen it somewhere in the question, and it is very, very, very crucial. Isoniazid resistant don't produce catalysts at all. Yeah, I think that's what we said previously. We said that isoniazid resistance don't produce catalysts at all. So it means they never produce catalysts. Whether it is resistant, whether it is catalyst, whether it is a heat stable or something.
Oh yeah, now it's making sense. Okay.
Yeah. So that is what happened. So they don't produce any kind of catalyst. So that is it. Mhm. Yeah, but catalyst is a good tool to identify to identify mycobacteria. Of course, when you want to look at catalyst production, and then you heat it, you heat at 60 degrees, then you will know that this one is not for TB. 2080 is also detergent. Detergent. So some bacteria produce 2080. I mean, hydrolyze 2080 in 3 days, others in 5 days, others in 10 days. It is just a detergent. It is like substrate. So some of them can hydrolyze it, others don't. It is every mycobacteria which produce lipase can hydrolyze 2080, but it depends on how much lipase is produced. So lipase can be produced, it hydrolyze it in 2 days, 3 days, 5 days. So it depends. So you just report it as it is. So we have seen how, for example, I mean not MTB, M bovis hydrolyze 2080. Not sorry, Mark complex, not bovis. Mark hydrolyzed 2080 in 3 days. So then we can also use it for that identification. So ion uptake, so we can use it only for rapid growers. So where we can differentiate, colony is rapid grower, so it doesn't uptake iron, whereas others uptake, and everything will become rusty brown. After iron uptake, it becomes rusty brown because of ferric ammonium citrate. After adding something like a ferric ammonium citrate, and they uptake iron, so everything become rusty brown. So then it means it is only used for rapid growers, but not TB. Can't use it for TB. RS alpha test is also good. Is another enzyme test that is used. So, for example, some bacteria which produces this enzyme include fortitam complex, coronary complex, xenopy, and then trivillae. This one, but it is not used at all for MTB. Pyrazinamidase, it can be used for MTB, yes. Yeah, but pyrazinamidase activity, which is used to differentiate. So because MTB can grow in pyrazinamides, it doesn't produce pyrazinamidase. So, but M. bovis can. So M. bovis from MTB, and then marinum from Kansasai. So we can use.
So I think the difficult part about these organisms and stuff. The thing you can easily remember the principle and also how it's like everything about certain biochemical. But the organisms that are in there, find a lot. Even the previous organism which did something else, it's also coming in there. So
Yeah. I know that one. For sure. That is true. That's what happens. But at least that's why I'm going to send you something that like a reference document that you will try to look at, especially when you are trying to deal with it. So maybe you can say, okay, let me try to sort my, and I advise you, and that's my advice I can give you, like what I used in my exam and helped me for sure. What I did was that you, after reading everything, you can give yourself like 2 weeks, 2 weeks or 3 weeks. Okay, you can say, okay, let me read microbiology in 3 days. And it's possible to finish microbiology in 3 days. You just like pick most important information. Then after 3 days, you take hematology for maybe 2 days. You also take 2 days. Maybe you cannot read the whole day. Maybe you can take like 3 hours per day. Read it. Hematology is not large. You finish it. Then you take another part. So it is simple. As long as you know what is difficult in a course, you can be able to go about it. Maybe you can say, okay, let me try to at least know some essential reactions for bacteria. We have talked about MTB so many times because I know MTB keep coming. Marker complex keep coming. Cancer site keep coming. Bovis. Others may come so not less frequently, but those ones keep coming. So when you know that M. bovis rect like this, cancer site like this, mark complex like this, MTB like this. So that will be enough. So some other things would be like some exceptions, like xenopy, high temperature. Marinum, you have to add something like mycobacterium. Hemophilus, Mycobacterium hemophilum. So maybe you have to add hemine. So those are some of the exceptions. But otherwise, uh, don't try to know everything, but at least there are some key things you have to go with when you are going for exam. So we are trying also to go through these principles so that if you know twin 80, what is twin 80? It will help you also to memorize that particular twin 80 for a bacteria because you know what is it. So that is, yeah, but don't worry, just you try your best. Microbiology is like that, but it's not like other courses. Other courses can be very simple. The same as tellurite, which is used for MAC. It is specific for MAC complex. So tellurite, positive. This is like to reduce a metal, tellurium metal, from colorless to black. So it is only used for MAC complex, tellurium or tellurite reduction. Urease is for good for M. scophylacum, which can be differentiated from Gordonei. Yeah, you may not see it. This urease test, urease, it is not easy to see it for MTB. If it is MTB or bovis or MAC, they don't even like talk much about urease test. It is not useful for those bacteria because we have most useful tests you can use for that. We have pyrazinamidase, we have T2H, we have niacin, we have catarase. So we have special unique tests you can use for TB, bovis, MAC, and so on. But like urease, we can't find it there. There's no need. Even if they are positive, but there's no need of that because we still have good tests for them. So urease is only used for scophylacum and Cotinea to help distinguishing. So when you see it, you can now try to remember scophylacum is positive and Cotinea is negative. Yeah, it is your exam time. So when you have an exam, then try your best. And also inhibition. So this is just when you want to use a chloramphenicol as an inhibitor. So some of them don't grow in chloramphenicol. So T2H is also something that you remember. T2H is used for M. bovis, which is susceptible to T2H. MTB grows in the presence of T2H. Remember that. M. bovis, T2H. T2H is very essential, very, very important. Sodium chloride tolerance salts. Growth on Maconchia, Fortitum coronae complex can grow. And something that is mentioned here is, of course, growth in the presence of hemine group, not hemine group, hemine molecule.
Ndasira pandanga so apa ndepena pangira muka 3 kwacha che. Stena
Okay.
then state okla na pesa paripe okla.
Oh, sorry.
Ndoneka sana wa choka fast mlandu.
Okay. All right. So that's what you can say. So I want someone to add something. That what do you want to add, or what do you want us to talk about next time? Because we're going to do microbiology in 2 days, 2 sessions. Maybe tomorrow, if we can, daytime. Because I want us also to have class, general class tomorrow. So, but before that, we can have this one. So.
There was a general question of which mycobacteria is transmitted, maybe through water, kind of. Is it coronary?
You mean the mycobacteria?
Yeah, there was mycobacteria, like which species?
Through water.
Through water?
All of NTM, all of them, soil and water. All NTM. Because NTM are acquired through water and soil environment. So they're not transmitted person to person. They are not.
Okay.
Yeah, so even avium, even cancer sign, so even xenopus. So all of them are transmitted like from environment, water and soil, yes, inhalation, but they are not from one person to another person.
Okay.
Okay. So tomorrow we can try. I'm going to see what time tomorrow we can meet before, because at least I want us to finish. Tomorrow we'll focus on gram positive, and then those ones that we call anaerobic, gram positive and the anaerobic. Then maybe something a bit about mycology. Then we parasitology is not something that can take our time. Parasitology is very simple. You just read parasites and know how it looks like microscopically. Because the parasite, there's no need of biochemical test, no culture, nothing. You just use microscopy and know how it looks like. Maybe with the pathogenesis. That's all about the parasitology. Okay, so let's close for now, and let's meet again tomorrow. I'm going to see. Today I was a bit busy before noon, so I was, but we
As
go
for
to
me, my only enemy is biochemicals.
yeah, biochemicals. Yeah. I've tried to mention how you are going to go about them. You just know them first and try to. I'm going to send you documents anyway. Let me send documents. Then you look at them. Each bacteria with each reaction, each
Yeah,
positivity. So then you will yeah, that one will help you. I'm going to share with you. Okay. Thank you very much.
yes.
Thank you very much. Let's meet tomorrow, and we'll see what's next. Okay. Bye, and see you tomorrow. Thank you very much.
Thank you.
Okay.